I'm Rosemary Medrano, and today we are interviewing Yayan Kiu, Senior Scientist of Molecular Biology at Genscript. And today we're talking about our new ITR platform. So what is ITR and how important is it during AAV production gene therapy?
Okay, thank you, Rosemary. And to answer that question, I want to start with the AAV. So adeno-associated virus, AAV, is a small single-stranded DNA virus initially discovered in 1965 as a contaminant during adenovirus proliferation. Recombinant AAV vector is generally replacing the wild-type AAV open reading frames with a target therapeutic or marker gene expression cassette. Currently, AAV viral vector is the most widely being used. ITR, so the topic, namely inverted terminal repeats, consists of an A to A primer, B to B primer, C to C primer, and D regions. This sequence form palindromic double-stranded T-shaped helping structure. ITR plays an important role in the virus life cycle, such as replication, amplification, episomal persistence, and integration. ITRs are the only site active sequence required for making recombinant AAV vectors, and also the only AAV encoded sequence present in AAV vectors. Intact ITRs are crucial for high titer AAV vector production.
Thank you for telling us more about what ITR is. So how does an intact ITR give better AAV yield?
Studies have shown that the deletion of different segments of the ITR sequence will affect the production of AAV. In details, the A to A primer region contains the rib binding elements, RBE, and the specific terminal resolution site, TRS sequence. RBE is involved in AAV genome replication, transcription, and integration. TRS is involved in AAV rescue and replication. The B to B primer and C to C regions also play a role in the binding of rep protein. Some studies have found D region matters in the AAV DNA packaging. Therefore, the intact RTR is crucial to AAV packaging. Before generating the virus, it is highly recommended to monitor the RTR integrity of the AAV plasmids by using restriction digestion and agarose gel electrophoresis. According to literature reports, less than 20 impurity is acceptable. However, several papers have confirmed that intact RTR sequence could lead to a higher yield of AAV. For example, the deletion of the B to B primer, C to C primer regions, or CPG motif of RTR reduced AAV productivity. When transferred to SF9 cells, complete ITRs are proved to obtain up to 40 more capsids and 10-fold reduction of non-AAV encapsulated DNA.
Can you tell us more about how ITR sequencing is different from Sanger sequencing?
Yeah, so because of the ITR its own structures, that it can form a very highly stable secondary structure, and the GC content is more than 80%. Thus, RTR cannot be sequenced through with traditional Sanger sequencing or NGS. To overcome this issue, Genscript developed a Sanger-dependent RTR-specific sequencing kit that can sequence through the 4 ITR with good signal.
And how does Genscript's ITR sequencing service and proprietary ITR strain stand out against competitors, and how does it accelerate gene therapy pipelines?
That's a very good question. So Genscript provides one-stop services starting from gene synthesis to virus packaging, including gene synthesis, plasmid production, gene codon optimization, mutation library construction, and virus packaging. Firstly, internal studies had shown that the success rate of cloning intact RTR plasmid from commercial strains is about 40 to 60%, such as NEB stable or stable 3. Our proprietary ITR strain, gene ITR V1, can increase that ratio to 90%. In further, this strain is adapted to the high-yield plasmid fermentation process developed by Genscript as well. Second, Genscript sequencing capability is more powerful and the QC standard is more stringent. Under the same standards, Genscript ITR sequencing pass ratio is 87.5%, while the competitor is 37.5%. In particular, Genscript can use nanopore sequence to directly examine the ITR integrity. Third, Genscript has extensive experience in cloning and plasmid prep of all kinds of cell type ITRs. Meanwhile, we also provide compliance service for gene synthesis, sequencing, and plasmid preparation to meet IND application. On-site audit is supported as well. To sum up, the integrity of the ITR is crucial for downstream viral packaging titer. Genscript's ITR sequencing, priority strains, and fermentation process ensures the ITR stability and high quality of plasmids as a core material, accelerating gene therapy pipelines.